not a purely signaling. But today I'm gonna just focus on something I have been recently pretty obsessed with, which is foraging, but because it's a school, I'm gonna present work that I might not agree with, but it's a very important background. So one thing I'm very interested in is the problem of decision making. So if you wanna simplify, decision making, you can think of something really complicated. The word have many states, you have to perceive the word, you have to do some computations in the brain, then you have to take a decision or a motor act and then evaluate your options, and then so on and so on. So how can we study, or in system neuroscience, how did people think about this problem? So we can simplify this figure by saying, okay, there is some psychological process happening, and simplify it as I have some sensory processing, perceiving the word, and then I do some computation, take a decision, and store some things in my working memory, and then I execute it. Okay? So I can lay out some psychological process and try to find a neural correlate that happens in the brain. This reminds us of Dan Katz's talk, and this is the approach that he critiqued. But nevertheless, there is very important work that was done in this area that one should know. So what type of decision making I want to focus on? So I want to focus on a very basic operations that happen in decision making, which, okay, I have the word have evidence, and then I accumulate this evidence and then take a decision, okay? So I accumulate decision 4, or against some decision. And it is, you can think of it as the hard decisions are usually slow and the easy decisions are fast. And this framework has been applied to all kinds of decisions. So let me just draw this down here. What is this evidence accumulation? So here I will go against a bit what Randall said in the morning. I will really be very abstract in terms of there is some cognitive process. So what we want to do is we want to say, okay, let's look at what kind of abstractions are being used by the brain in order to do something like evidence accumulation. So you can, if you have, you can draw something like this, okay? And then I have something called an abstract variable called the accumulator in my brain, begins at 0. And then I am receiving some evidence. So, and then, okay, I will keep accumulating till I reach a certain threshold. And this threshold is, once I cross it, this is a decision. Okay? But I can go slower. Okay? So this takes more time. So this is a harder decision. And this is an easier decision. And it might be, for example, if I have a binary in a two-forced choice task, I might have right or left decisions. Okay? So this is what is called noisy evidence accumulation, and that's exactly what I have drawn here, exactly. So yeah, and then here, this is a decision, this is what we call decision border at 0, okay? So this is a very good intuition for this type of evidence accumulation processes, okay? Cool. And this has been studied for a very long time in both psychology literature and in the system neuroscience and literature. Okay, so if you're a system neuroscientist and you want to study this, so the first studies happened in monkeys. So let's do some monkey anatomy. So this is the brain of a monkey, and then you have many brain areas. Some of them are visual. So here, this is visual cortex. Here, I will not bore you with all the names. The lateral anterior parietal cortex. You have something called frontal eye field and prefrontal cortex, and area IT. Okay, so what happened is that these experimentalists were able to train monkeys to do something like this evidence accumulation. How? That you have the monkey sitting in a chair, and then they have a screen in front of them, and then they have dots motion in front of them, okay? Like, let's say you have a stimulus, you have dots, okay, moving this direction and others reverse direction, and then you want to see what is collectively both of them are moving. And then you can vary the difficulty depending on the coherence of the stimulus, okay? And so you can simulate here the easy and the hard trials. And again, that's basic knowledge that I am presenting to you here is you can, from these experiments, you can do, sorry. Oops. Okay. One click. Good. So you can draw something like this psychophysical curve to know, okay, how do you know that the monkey is doing the task? The monkey, okay, well, how you know that the monkey did the task? It will saccade, for example, let's say the collective motion to the right, it will move its eye to the right, you know, okay, that's a trial, correct, move on to the next one. So you can get a reaction time, which is how long it takes them to solve this task. And then, okay, but because monkey, I cannot talk to the monkeys and tell them, okay, did you do the task? We know this from looking at the psychometric curve, okay? If you plot the motion strength to the percent correct, okay? So you get this at the easy trials, you get 100%, and the more difficult, your percent correct decrease, okay? And the slope of this curve tell you how good they are at doing the task. The revolution in the field happened is that when they stick electrodes in brain areas, we don't need to go specifically which brain areas they use, but here, for example, in area MT, they put electrodes first in LIP and then MT. So LIP, they recorded during the task, and they found that the neurons in this area ramp at different slopes depending on the difficulty of the trial. So as if this neural process is basically mimicking this integration processes happening here. They recorded from another area called MT, like this. They found that it doesn't increase, like have this ramping activity, but it has a more sustained activity. Usually when you see more sustained activity or persistent activity like this, this is indicating that this might be a memory area, because it's just, okay, reverberating activities that may be maintaining some variable. Another thing, another area they recorded from is the FEF, and this area seems to be active just before the animal move its eyes. So this is what we call pre-motor activity. I think Daniel mentioned pre-motor activity. So what you see here, why this tasks have become very important, very foundational in systems neuroscience, because of this type of, okay, I can record from different brain areas, and I can get into different aspects of the behavior. The issue with monkeys, at least in US and in Europe, is that you can only have 2 monkeys. It's very long. It takes a long time to train them. So many people have been switching to study this behavior in rats, because you can use much larger number of animals. So you can do the same with rats, but in this case, so one assumption underlying this evidence accumulation, it is invariant to the sensory modality. So let's say we did it with the visual stimulus with the monkey, but here with the rat, we do it with auditory stimulus. So the rat enters inside this behavior box, and then this behavior box have three ports. The animal will poke in the middle, okay, and then keep standing like this, and then we'll hear clicks on the right and on the left, and then at the end of the trial, we'll orient towards the side that have the highest number of auditory clicks. Okay, so here we translated the behavior to the auditory domain, and then we can repeat this, but then we can really do like a psychometric curves from a large number of animals. So the previous plot, this was average of like 5,000 trials, but here we have 19 rats, and every rat have around 20,000 to 100,000 trials. So that's a very large number of trials. Okay, so we can get better statistics, because rats, you can have them in abundance. And Okay, so this is called the psychometric curve. Another thing we can learn is something called the chronometric curve. So what is the chronometric curve? If you hear this word, this means that, okay, one thing I didn't tell you is that, for example, the trial length, when you design your experiment, you don't want the trial, all the trials, for example, to be 1 second. Why? Because over time, the animal can develop a timing strategy so they know when it will begin and when it will end. So we trick them by, for example, having 100 millisecond to 1 second trials, and it's a Poisson distribution, and you keep randomly sampling from it the trial length. So, and the accuracy of the animal, the performance of the animal will depend on the trial length. So here, let me explain it slowly, is that the difficulty, again, let me repeat, the difficulty here is the ratio between the right and the left side. Okay? So if I have 39 clicks at one side and 1 click and so on, this is a very easy trial. So immediately, okay, I go to the side that is 39. But let's say I have this 26 to 14, which is much more harder, so it will take me more time to integrate, and my accuracy will be much less. And the accuracy will increase with the increased stimulus duration because I have more time to integrate. Another difference than the monkey task is that usually you will hear people telling you, okay, this is a reaction time task. So a reaction time task, I don't give the animal a trial length, I just wait till they do the task and then close the thing. This is harder to do in rats. We can discuss this in the questions. Okay, so back to the model that the abstraction I have drawn here. So how do we know that the animal might be using this? So how is the animal solving this task? So we can formalize this process here using something called drift diffusion models that some of you might be familiar with. Today there will be a lot of math in the talk, but the important thing is to get the intuition behind the models, and in the readings I have put the papers that have all the derivations. So let's again repeat the story. I begin at 0, but now I have this variable A, my accumulator, and then there is a trajectory that's evolving, but it's a stochastic process. So I have many sources of noise. First, I have an accumulator noise in my accumulator. I have a sensory noise, which is how I am perceiving the environment. So let's say the rat is receiving this auditory click, okay? You know, if it is a perfect word, the amplitude will not be modulated. I will perceive it like this, but usually there are two things that will happen. It will be jittered in time a bit, and it will be reduced. So there is two processes here happening, which is the sensory noise that jitter it in time, and this is a Gaussian noise, or there is an adaptation process, okay? There is another thing here. You might think, okay, is this process perfect? No, this process is what can be leaky. Okay, in a perfect world, it will not be leaky, integrate faithfully, but usually there is some leak due to, you know, that the process is not perfect, and we can quantify this using this lambda parameter, okay? And then you have the threshold as a boundary that you kick, and this is called drift diffusion model. And then you can write it down as a simple equation where you have here your accumulator, and then you have your adaptation parameter, and then you have your clicks on the right and the left, and then you have your noise term and the leak term. Okay, very nice. So that's a way by which we can mathematically model, okay, what the animal might be computing in its mind. You can also tell me, okay, but what this behavior you have presented is very simple because, oh, this is a stationary environment. So let's add something a bit more complicated. So this is a very nice paper by Jack Kilpatrick, a collaborator of mine for a long time, where he generalized the drift diffusion models to include dynamic environment. So what is dynamic environment? So here, when I talk to you, I say, okay, that the word is not changing. But what, let's say we want to approximate something that happened in the real world, the statistics of the word might be changing. So how does the animal deal with this? So we trained the rats, in this case, on something called the dynamic click task. So what we did is, instead of just saying, you have stable auditory clicks presented over time, is no, that will change in the middle of the trial. For example, I have 4 clicks here, 1 click, and suddenly it switched to 4 and 1. Okay, this is the simplest thing you can do, and then you can define something called the hazard rate, which tell me how fast this environment is fluctuating. And so, okay, the optimal strategy to solve this will not, if I keep accumulating without all the time, I will just miss the switches. So the animal need to optimize in order to discount or throw away some of the evidence it is receiving in order to be able to keep track of this state change that's happening in the environment. Cool. Guys, you can interrupt me if things get very crazy. So, okay. We can go and say, okay, let's try to fit this drift diffusion models to this behavior. And what is the prediction? The prediction would be, okay, in the static environment, usually lambda, this has to be around 0, okay, because I have to perfectly integrate. But if I am in a dynamic environment, this has to be in the negative, okay? I have to throw it down. So we fit the models, and then we find in the static task, we have it around 0, but it is not very well controlled because it is, imagine, I'm just leaving the animal for a long time with static environment, so you cannot really control it around 0. But then when we have this changing environment, for example, this data from a hazard rate of 1 Hertz, we are able to get this negative lambda, okay? Just to get the intuition is that using this type of modeling, we are able to say that the animal might be using a leaky integration computation in order to deal with this change in environment. And then we can play all kinds of games, like have them in this changing environment and then to a stationary environment. So you are able to really change this lambda over time, which is something, tells you something about the flexibility of the animals, okay? So, of course, I didn't mention foraging till now, so you might be wondering what's happening, but that's a very important introduction. So then in the middle of my postdoc, I got this like existential crisis. Okay, what the hell am I doing? So there is something really hidden in what I said is how long does it take to train these animals? So on average, let's say the first task, it takes around 4 months, and the second one is 6 months. So that's a very long time, okay? So, and then it made me, I began to think about, okay, the whole framework of animal training. So the whole motivation is to be able to control the animal, because if I have the animal running around, okay, no, we need to have repeatable trials because this is how we control the behavior. And then afterwards, you can stick an electrode and try to relate some neural recordings to the task variable. But then there are many caveats with this. So it takes, 3 to 6 months average time. Another thing usually people don't tell you is that, okay, you have to really restrict these animals from motor in order to motivate them. Because, you know, rats in their everyday life don't do this type of task, you know. It's a very strange environment for them. Another thing is that are we just training the behavior or also the brain? Another thing is the state dependence, you know, like many of us, okay, I wake up in the morning, I'm in a good mood, I want to, or in a bad mood, I want to do the task, I don't want to do the task. But what is expected from the animal is that every day they have to perfectly perform the task. And the body, okay, so posture, size of the body, what's computed. So what have been happening for a long time is that specifically systems and cognitive neuroscientists, they are dealing with the behavior as if they solved the brain in a vat problem. So as if inside this behavior box is only the brain doing the task and you don't care about anything else. For a very long time, I was not even, I don't see the animals. I just get trials and clicks and don't even see the animals. So okay, I kept thinking, okay, what do I do? Do I just give up on this whole thing and go back to physics, or what do I do? So in Woods Hole, if some of you might know it, it's a place in the United States that have a lot of very good courses. I met my longtime collaborator for 13 years, Jacob Davidson. And then over some, my favorite whiskey, okay, Talisker. So we kept thinking, okay, what decision-making tasks can we study that is very close to the animal? In the same time, in the same course, I got to meet Emily Makovich, who became my collaborator, and Ralph Peterson. And then, meanwhile, I was also living in New York, and then this is my favorite thing now. So you have these rats running around everywhere in New York City. It's a bit slow, but you know, the pizza rat, that's very famous. But I took this video, but you know, you can see this everywhere. And currently, we have a project running where these are, for example, recording of ultrasonic vocalization of these rats in the subway. We have thermal cameras and other things. Okay, so that's have been a big obsession of mine. I like these rats. They're very smart. They adapted to a very harsh environment like New York subway. And yeah, nobody can get rid of them. So yeah, you can read about us, because also we were covered in the media. So okay, what is the name of this behavior? Oh. Foraging. Okay. So so what is foraging? You know, it's animals have to acquire energy, then they have to search for food in order to self-sustain themselves. It's universal, so all animals do it. I cannot think of any who don't forage, find food, acquire energy. It happens over multiple spatiotemporal scales. So you can do a little foraging experiment in the lab, in a box, or you can study bats that are flying, birds, or monkeys in a forest. So it happens over multiple spatiotemporal scale. It's sensitive to the environmental structure, so you have many animals, because many animals forage in different conditions, so animals have adjusted their foraging strategy depending on the environmental structure, and it has a very long history of being quantitative in behavioral ecology. And you know, I took a deep dive into foraging theory in behavioral ecology. And recently there have been a strong interest in foraging in neuroscience, you know, as a decision-making behavior. So how do I think about the problem of foraging in my group or in my research within my collaboration network also? Is, okay, let's ignore that these are bats. I had this for a grant. But yeah, animals usually have their own private information, so they get information from the environment, and they also have some information from their conspecifics. And then they have to take some movement decision toward a patch or a resource of food. So how do we dissect this problem? So you can ask the ecological question about the fitness. So how this particular foraging behavior contribute to adaptability, life expectancy, mate choice, reproductive success. We can also ask something about the behavioral algorithms, like how does a single animal take input from the environment and integrate its own foraging experience, its own internal state, social information. How does it inform or how this computation is happening to the foraging decision? And you can ask a question about the neural implementation of this behavioral algorithm. So that's how I think about this problem. So today our focus will be on the behavioral algorithms, and I will try to show you some of the progress we have done in this area. So let me just. So foraging behavior is a very wide, type of behaviors, but the very specific behavior I'm very interested in is called the patch foraging behavior. So if you have an environment like this, and then it has some resources, and then, some animal here, sorry, my artistic skills are, will go inside the patch, consume some food, and then leave and go next to the next patch, It might look easy, but it's once you dive, the richness of the behavior begins to unravel. So you have here behavioral observable, which is first the patch resistance time, like how long does the animal stay inside the patch. And then you have the travel time, the time it takes to travel between the patch. And then you have the amount of food you consume inside the patch, which is the food intake. Okay? Cool. Okay, so how to think about foraging? So, okay, I have this problem. I was sitting with Jacob, and then, okay, what do we do about it? So, Sisyphus, yeah, it's very good. So, let's develop some models. But it's very important to realize that modeling is basically a very Sisyphusian activity. You know, develop a model, you keep working on it, and then it encounters the data, then it falls over your head, and then you go again. But important thing when I present the models here, I don't take them too seriously, first of all. I use them as a way to design experiments, analyze data, but I don't have any like emotional attachments to them. It's not that, you know, I have a model, I want to prove that it happens. But it helps us generate hypotheses, develop ideas about how to understand this behavior. Because you can see if I want to do neuroscience on this, if I'm moving from this paradigm where I have a highly trained behavior into a place where the animal is moving around, going from one resource to the other, how do I use some of the quantitative tools that have been very useful to understand the trained behavior in this type of natural behavior? And this was our point of beginning. But let me zoom out again. Already, as I said, the behavioral ecology already have done a lot of quantification under what is called optimal foraging theory. There is a very famous theorem called marginal value theory, which assumes, okay, if you have the environment is divided into patches, the animal will leave the patch when its current patch intake is equal to the average environmental intake rate. And you will usually see, and this is it's pretty old from the 70s, you'll usually in many behavioral ecology papers, you will find people plotting time foraging in patch to the cumulative resource intake in order for you to get this optimal time in patch. But if one look closely into the theory, you will find very strong assumptions. That the animal have to know basically everything about the environment. The current food arrival rate, that the food is arriving continuously, that the environment is not changing. Have been used heavily, have been very useful, but it's another important thing, it doesn't tell me any mechanistic insight. It's a rule, it doesn't tell me how the animal will implement it. So throughout the talk today, I will use MVT as a guiding principle measure for like optimal comparison. So like we do with any modeling, we begin by asking mechanistic questions. So usually, okay, this is how I approach modeling, I begin to ask questions. So what the questions we were interested in is first, how would the animal implement something like this marginal value theorem rule? Sure.
Just to be sure that
No, you need, yeah.
Just to be sure that I understood exactly what the marginal value term says. So this is like a average theorem.
Yes.
So it's about the average whole population intake would be equal to how much actually the environment will receive on average. That's the idea.
Yeah.
Okay.
Yeah. Okay. And the assumptions that the animal will, that's how they leave the patch.
But doesn't tell you anything about in a single trial or the single animal what's going to happen. Okay.
No. So that's another thing that what happened when we begin to check, because the interest in ecology or theoretical ecology and population ecology is very different than, it's more on average, because you don't want to know what's happening on a single trial with animals, but if you want to study physiology or neuroscience, then it becomes, you need to know this about inter-individual variable. Okay, so what happens if we have uncertainty in the environment or the foraging process, but how does the animal learn the structure of the environment? But something else very, very important is that animals rarely forage alone. So and this theorem doesn't tell me anything about what happens in the social context. So in the remaining of the talk, I will try to answer some of this, and as you can see, once I began to work on this problem, it was like a dark hole that just sucked me in. from 2019 till today, and we published a lot of papers trying to answer these mechanistic questions. And I will give you a flavor of some of the modeling approaches we use to answer every question that I posed here. The first thing that we did, before I go to the, is to say, okay, let me take what I already know and try to map it onto foraging. So let's say I am the rat here. I enter into a patch, and then I'm eating some food, and I try to imagine what's happening in the rat brain. Again, we go back to the accumulator formalism. I begin at 0. I receive a piece of food. Then this piece of food will act as an evidence, and over time, I take another piece. Over time, there will not be food. I cross, and this crossing will be considered leaving the patch. Okay. So this led us to develop what we call the foraging drift diffusion model. So I begin by something that I'm already familiar with, and said, okay, let's take this models that have been successful in decision in psychology and decision neuroscience and try to extend it to the foraging paradigm. But the nice thing about foraging is that you can, vary many things in the patches. So you can parameterize the amount of food, the patch size, the food chunk size, and the travel time patches. But you get the gist of it. And then you can write this down in terms of 2 equations. So here, as Daniel, you will see this. Daniel pinpointed something very important. Ecology you see from the big environment, averages, but here I try to take the perspective of the animal, how the animal is doing a certain computation. So you can write this in 2 equations. You say, okay, there is one thing that tracks the change in the energy in the environment, okay? And the second equation is basically the same as the previous model I presented, is the decision to leave the patch, which is, I have a decision variable here that have a time scale of update, and I have a drift rate, that's a very important parameter, and then have some reward rate, which is the rate of food intake, and some noise term. And you leave the patch when X reaches a threshold. Okay, we will see now how we compute this threshold. But if you simulate this too, you write down the Fokker-Planck, blah, blah, you look at the full probability distribution, and you get this very nice, probability density evolution. So of course, I here simplified it. You usually don't have like single trajectories. Usually what you simulate using the Fokker-Planck equation is an ensemble of trajectories, so you have a probability distribution that's evolving over time. And you can compute it exactly in this case. Another thing in the modeling, usually, I'm trying to get you into my head how I think about problems, is I try along with my students and collaborators to begin with first with the analytically tractable models. So any model I'm going to present it today is fully analytically tractable with extensions that we can numerically simulate. Okay, so save you some time. This threshold. So you can say, okay, well, what is this decision threshold here that I have to cross? So the decision threshold depends on many parameters. First of all, you can imagine on the patch size, okay, on the drift rate, on the amount of food, and the average energy in the environment, plus this metabolic cost. So there are things here I need to define. So metabolic cost here, I will keep it constant. But we will relax this assumption later, but you can imagine that if the animal is moving for very long distances, that there will be changes in this metabolic cost, it's burning energy, other things. And the total energy in the environment, you can imagine it as some kind of like the entropy of the environment, given some amount of food. But the interesting thing, okay, so we have this integration strategy. And this is a heuristic that we assumed, okay, the animal might be doing. Do we stop there, or we try to think, okay, what strategies, is there other strategies that the animal might be employing? Because you can think there are a couple of things happening. I can enter the patch, for example, and try to estimate the density of food. So I do some density estimation. Or I can enter the patch and try to count the amount of food. Okay? So see, I try to think about problems usually intuitively and then go back to the blackboard. Okay, so we found if we plot the drift rate with the threshold, we get these two classes of strategies, what we call the increment-decrement strategies and decremental strategies. So let me show you how they look like. So what is the increment-decrement strategy? So the increment-decrement strategy tell me that if I find food, I'm more likely to stay in the patch. And this happens if your drift rate is positive and your threshold is positive. So you can just, again, intuitively understand it that if I get a piece of food, then I'm more likely to stay in the patch till there is no food anymore, I just leave. Another one called the decrement strategy is finding food makes the agent or the animal wants to leave the patch. So I count 1, 2, 3, 4, and then leave. Happens when the drift rate is negative and threshold negative. You might be wondering, okay, but are you artificially constructing these scenarios? No. We can actually, one thing I didn't tell you about is what does the animal know about the environment? So if the animal doesn't know the amount of food inside the patch, so there is an uncertain food, patch food density, so of course, when I enter into the patch, I find food, I'm more likely to stay in the patch. And we can look at it, for example, you can have an experiment where you try to increase the patch food density, plot it to the patch residence time, and you find a quasi-linear relationship between them. But let's say I am in a familiar environment where I know that, for example, this patch have 4 pieces of food, so this will be the counting strategy. So I count, there is 4 pieces of food, I count 1, okay, I know that there is 3 remaining, so I'm more likely to leave the more I take food. This sounds trivial, but we can write equations for this. So we have this heuristic, and then we have some variety of strategies that depend on the threshold drift rate. But one of the reviewers of this paper told us, okay, but can you develop then a more normative approach to derive these decision strategies? And this got us into the Bayesian approach to patch foraging. So what is Bayesian approach? What is Bayes? Bayesian learning? Yeah, what is Bayesian learning? Exactly. Very good. So that's a statistical learning. Okay, so means I accumulate evidence about the environment, and then I do some kind of inference using the statistical evidence that I accumulate. And there have been, a lot of papers in behavioral ecology trying to see whether animals are able of Bayesian updating, Bayesian learning, are they Bayesian foragers or not. This paper is nice. Bumblebees learn to forage like Bayesians. But the drawbacks of the models in behavioral ecology, as I have mentioned just to Daniel, is big averages. The parameters are very abstract. I will not be able to relate it afterwards to body posture or motion data or neural dynamics. So what do I mean to formulate the patch foraging problem as a statistical inference problem? So let's think about what problems does the animal have to solve. So let's say the animal falls from the sky into an environment, so it has to find first a habitat. So the habitat is like a forest, okay? So I want to find a good forest, and this is called the habitat choice problem. And then once I find my habitat, then within it, I begin to do this patch exploitation. Why did I divide this into like this hierarchy of problems? Is that the habitat choice, so you might, if we go back here, if I have a patch of food, this patch might be depleting or non-depleting. So depleting means the amount of food is decreasing over time. Non-depleting, it never ends, or it depletes over a very long time scale, like a forest. So the habitat choice can be approximated by a non-depleting patch, and the depleting is the case of this patch exploitation. So what does a statistical inference problem mean? Is that the animal, for example, have a prior over how rich the patch environment is, and then go into a patch, begin to sample the food inside the patch, and then they encounter the food, and then use this encounter in order to build observer belief. And then you can write this in terms of Bayes rules that I'm sure a lot of you have seen it. And this is just the first intuition. Okay, so what happens when we have this habitat choice problem? So let's begin by the habitat selection problem. And what we usually do is we want to find what behavioral algorithms or strategies the animal might be implementing. So let's say we go to this scenario here. We have a habitat where there's 3 non-depleting patches, one that is very rich, one intermediate, and one poor. And then you want to ask, okay, how would the animal act in such an environment? So let's think together. So first we would think that the animal will keep searching, and then at the end will remain and stay forever in this high yielding patch. Okay? So our intuition tells us that the animal will try to minimize the time to find the high quality patch, and the more obvious this high quality patch, this will strongly shape the animal behavior. So let's see if this holds in our models. Good. So very important to introduce you some of the models that haven't been introduced, and we have treated more complicated cases, but I want to begin by if we have a binary environment. So let's say a high and low environment, and it has some lambda h, lambda L, and with some, it exists in the environment with some probabilities, okay? So what you can do is you can write down a partial differential equation that tells me how does the belief of the animal, the internal belief of the animal, evolve over time, okay? And that's basically, this is the log likelihood ratio that take into account the food encounter, and this is, this difference is the lack of food. And the best thing is to see, and if this is positive, this tells me that my belief suggests that this is high patch. If it is negative, my belief suggests that this is a low patch. We can look at it graphically by seeing, okay, if we plot the belief over time, let's say I begin in the high yielding patch. Okay, I'm in the high yielding patch, then boom, switch to the low, go to the high, switch to the low, tack, tack. I keep doing this, going back and forth. Still, asymptotically, you will leave the low yielding patch and stay forever in the high yielding patch. So what we are proposing here in the best strategy is to arrive and remain in the high yield patch as soon as possible via a sequence of what we call stay and go decisions, okay? And then this, we can compute a departure threshold here, which correspond to the time at which I realize in my brain, oops, I am in the low one, let me exit and go to the high one. Calculating the first passage time problem for this, we can get this ugly expression, but it tells us this is what we are trying to minimize here. That's arrival time, okay? It depends on the escape probability from the high yielding patch, okay, and the time to exit the high yielding patch and the time to exit the low yielding patch. So, very nice. So let's play with the parameters a bit. So what we are trying to do is to minimize the arrival time. So let's play in our simulations and plot the arrival time, which we can compute exactly here, and keep the low yielding patch constant and vary the one with the high yielding parameter. And then the more we increase it, the more we minimize this arrival time. The dots here are the simulations and the curves are our analytics. We can also keep the ratio of the high and low patch constant, and then vary how probable is to have this high patch in the environment. We increase it, we minimize the arrival time. And so this, if you think about these parameters, all of them are observables from the experiments. You can know how early they arrive to the patch. You might know how rich the patches are. You can compute all of this. The thing that these models offer us is to be able to compute this optimal threshold. And this optimal departure threshold, basically, we can compute it by integrating the previous equation by looking, okay, if we increase this high yielding and if we plot it intuitively, if we increase this high yielding, the high yield rate, okay, our optimal threshold decrease because we are able to accumulate evidence much more optimally in the environment. Great. So the behavioral algorithm we are proposing here is that the animal is doing a log likelihood ratio computation of the high and low environment over time till it crosses a threshold. And the interesting thing is that we didn't even talk about reward rates at all. So the animal, usually when an animal is in a task, they say, okay, you try to maximize your own internal reward rate. But here we talked about just minimizing the time to find this high quality patch, which is a very strong deviations of the marginal value theorem. Because also the marginal value theorem hidden in the details, it assumes that there is, you are dealing with a depleting patch. And the more obvious this high patch, okay, this will strongly shape the time and strategy to find the high patch. So what if we change a single parameter in these patches and make it depleting? So the patch exploitation problem. Do we expect the animal to behave in the same way? Obviously not. So if we just change the patches from non-depleting to depleting, what we can think about, okay, in this case, the animal will keep moving between the patches, deplete a bit, and depart, deplete, and depart. And then over time, it will keep spending more time in the high-yielding patch. And of course, asymptotically, the whole environment will be depleted. So if there is no uncertainty, if the animal knows what the yield rate of the current patch, so this will totally obey the marginal value theorem. But if there is uncertainty, so the animal, for example, doesn't know how rich the patch is or uncertain about the richness of the patch, the forger strategy will deviate from the predictions of the marginal value theorem. Okay, so here we are back to familiar territory, which, okay, in this case, because the animal have to compute what the underlying reward rate in the patch is, the strategy is to optimize the long-term reward rate. And as I said, if the animal knows everything about the environment, okay, the animal will be able to optimally get the optimal exit times that corresponds to the marginal value theorem. But the most interesting part is to look at the uncertainty. So how can we model uncertainty here? So let's say I have the animal have 2 patches and one have 5 pieces of food and one have 1. Which one of them is more uncertain than the other, statistically? If I enter and try to search, which one is more uncertain? No, you don't answer. If I have 1 and 5 pieces of food and I'm entering and searching inside, which one I will have higher uncertainty? Answer. I want an answer, yes. This one? Yeah. Exactly. Okay, great. So that's how we model uncertainty. So there is many ways to model uncertainty that we had in our study, but one of them that I think is easier to explain is if we change the chunks of food. And basically, this is what we find. Okay, so if we decrease the amount of food, the animal will keep deviating more and more from the optimal marginal value theorem exit time or leaving time. So we can see from this curve, this normalized curve is the optimal of the marginal value theorem. And then the animal will leave high patches too soon and stay in low patches too long. This effect is called overharvesting, and you will find a whole dozen of literature on overharvesting. Because basically, if you look at ecological data, it's very rare that marginal value theorem really holds. Usually animals are overharvesting. They stay longer in patches. So what will be another way to model uncertainty? To make the animals stay longer or no? Suggestions. I want suggestions. don't be worried. How can I model uncertainty more? No, don't answer. Exactly. Okay, so I might be, let's say, if I have a rat here inside the patch, and you think that there is a predator outside, okay? So this will make me stay longer here. I am very uncertain about leaving. And this is, you can, in prospect theory, if some people familiar with behavioral economics, there is something called the utility function. And this utility function, the shape of utility function tells me how, whether a subject is risk aversive or risk taking, and then you can do this quantification. So usually rats, for example, are very risk aversive. They don't like open spaces. That's why when you do experiments, they keep sticking to the wall because they think there is something will eat them outside. So this is a way we have it in our papers that you can basically distort the way they perceive the reward. Okay, let me eat more slowly, for example, in order not to leave early. So they stay longer inside the patch. So this is another way to model uncertainty. Okay? So again, it's very important to keep it intuitive. You know, the math one can read the papers, but it's very important to think about it intuitively from the perspective of the animal. And yeah, risk is a very important parameter. Another thing also, if we consider, as we are talking about prospect theory, there is something called, okay, so you can say reward maximization. So do I keep maximizing the rewards forever? Or I can plateau, and this effect is called satisficing. It was proposed by Herbert Simon. Okay, so maybe the animal has some internal, like kind of a plateau in order to plateau at. And this effect is called satisficing. Maybe you have too much food, but they will stop eat at some point and leave. Okay, so it's very important. So this is a lot of things coming from economics, and we try to include it in this type of decision making. Yeah, you can, you know, if you go to Wikipedia in the page of biases, there is so many biases that have been reported. Hot hand and, you know, all kind of, mostly BS, but biases. But the important thing, I'm just mentioning Herbert Simon satisficing and stuff, is just because of the notion of optimality. So usually you'll say, okay, this is MVT optimal. Really, animals will behave optimally, but usually this optimal rule is very important for just comparison. Because you can say, okay, the animal is behaving suboptimally, but in what way? Is there a systematic way by which they have this like suboptimalities? Okay. So in a depleting environment, what the animal is doing in not likelihood ratio is trying to compute a maximum likelihood. Do people know what maximum likelihood estimation is? Okay, can somebody explain to me? Yes, you. Yeah, explain. No, behind you. Yes, you. You said yes. What is maximum likelihood estimation?
Yeah, so it's to determine the probability distribution.
Exactly. Exactly. So I have a probabilistic measure, and this is what I try to do. It's very important to tie to the next part. So here, just by changing a single parameter in the patch, we totally propose that the animal will be using another strategy that is closer to marginal values here. Okay, so we keep moving. What time is it? I don't know. Good. Yeah. What? Yeah. Less than this. So, okay. So, but then the treatment I did now assumes that the animal knows the environment, learns the environment. So how does animal learn to forage? And this brings us back to the maximum likelihood estimation. So let's say, how do I learn an environment? So if I oops. I think it's nice. So you have, let's say, a couple of patches, so you don't know the environment at all, you are the stupid rat here entering, and then begin to sample. So you do some sampling, and over time you build a probabilistic image of the environment. It's never a deterministic, measure. We might, in the discussion, we might, discuss this, but I believe that, stochasticity go down to the most fundamental level. Okay, so we keep doing, the animal goes do some patch visits, and over time, so here what you can plot is basically what's your name? What's your name? Yeah, like Felipe said, you compute the probability over time, so you can do this. Basically, this is the process you are doing. Over time, you can see how hard it is to learn when you change the richness of the patch, and you can also compute the max. Okay, what is MSE? Oh, what is MSE? Answer, mean square error over the estimation. So basically, like you do your calculation, so you assume, okay, the animal might be doing also a calculation like this. This is a non-depleting case, and then this is the depleting environment. So can somebody guess, would it be easier to learn the depleting or the non-depleting environment? It is, the answer is in the figure, but somebody can tell me what would be your intuition. If I have a depleting environment and a non-depleting, yes?
I think it's the non-depleting, because the depleting you need time to learn the rate. There's more learning.
Great. It's nice to hear that someone understood. Sometimes I have. Great. Okay, so you learn the encounter rate. There is a very nice paper by a group at DeepMind where they took my model. I worked closely with them. And then, okay, they said, can we take this model but extend it to a very complicated, like spatio-temporal thing? So they put it in one of their black box deep RL agents, and the agent was able to do like all kind of learning a complicated spatio-temporal scheme. But the mechanism is basically a maximum likelihood estimation, but this is analytically computed. But then if you want to expand to, first of all, you notice that I never talked about space. So there is no space in this model. The animal, I do accumulation here, decide, then boom, jump here. But the space in the middle we will talk about afterwards. But if, again, because of my obsession of having exact models, but you can check this paper, it's really nice. You can expand it to more complicated scenarios using deep reinforcement learning. Then they have this robot in digital space, and cool. So just to recap, we have shown that we have this normative framework that allows us to propose some behavioral algorithms that the animal might be using across environments, either in the habitat selection problem or in the patch exploitation problem. We have shown that, uncertainty, one way to model it is by controlling the amount of food and really have systematic deviations from optimality, or we can also have some prediction about learning the food arrival rate, and as have been told, it's quicker in the depleting environment. So so now this is the single animals, and this is the first part of the talk. So now we want to extend it to social. So can somebody, if you have this model, how can you extend it to the social realm for two animals? Just two animals. What would you do? If you have this model in your hand, some drift diffusion model, some stochastic process, and you want to extend it to two animals, what would you do? Suggestions? Yes.
You need to keep in mind the other animal strategy, because you're competing for the same resources, so there's like more degrees of freedom around that.
Mm-hmm. How would I do this? How would I do this formally? Exactly. So you just couple. So you can have coupled stochastic processes. Okay, that's what we are going to do in the next part. And so, so let's first, before I go into the thing, is to think about the variety of ways animals might couple socially with each other. So first of all, I might have diffusive coupling. So diffusive coupling means as stochastic process is evolving, the other animal will follow this belief coupled with each other, okay? So we, as if like, let's say it's like mental, almost like a mental reading. And second thing, it might be, another thing is that I see that the other animal is in the patch, so I think, oh, this might be a good patch, let me stay with them. I see them eating, then I eat, that's reward coupling. And then if I have more than two, I see a lot of agents in the same patch, and I say, okay, let me stay there. This is a counting coupling. So we will treat each and every of these cases and see how do they change the dynamics of the model. So now, and we will compare this to the non-interacting age. So what does it mean non-interacting? So let's say in the basic scenario that we have more than, now it's more than two, so I have an n number of animals, and I have two patches, patch 0 and patch 1. Patch 1 is the rich one, and the poorer one, and there is some travel time. So if they are not interacting, there is no social coupling, you can write down the vanilla Drey diffusion model without any modification, and then you can simulate the group behavior as they stay more time in patch 1 because it's the richer, and then they go to the other one. The nice thing about this model is that you can compute fully the probability to leave the patch and the probability to be in the patch. But let's see how they look like. So the probability of leaving the patch look like this damped oscillation till it reaches some equilibrium. So what's happening is that the animals keep moving, maybe together and going back and forth, and then there is an equilibrium probability at which they are redistributed. They are distributed to both patches. Okay. We see the same pattern, of course, for the probability of to be in the patch. Okay. So that was the first finding that we found, is that if you have this a large group of individuals, they are moving together. So either they will, you can see, you can see that if the spikes keep going like this, there will be like a more cohesive group. If it goes very fast, then it's a less cohesive group. Okay, but this is in the non-interacting case. We can compute the equilibrium, the stationary probability. The most important parameter we focus on here is this: we have to have an effective drift rate. Okay, and effective drift rate is usually computed for every type of coupling that we do. But the most important thing we use for parameterizing is something called this gamma, is telling me how fast these oscillations are decaying. And a bit counterintuitive is then if this number increases, that means the group is more cohesive. Okay? So let's go back to the couplings that I promised you to show. So let's go back again to say what couplings can we model. So today, in interest of time, I will only treat the case of the egalitarian structure. So let's say we have an egalitarian society where all individuals are equal. So there is no explicit hierarchy. And we have these two patch types. So the animals might couple with a diffusive coupling, which means like we couple our beliefs together, or pulsatile coupling. It might have two signs. So one animal leaves, I couple, I live with you, or the animal arrives, I stay. Reward, I observe you eating, I eat. Reward coupling. Counting, I see a lot of animals in the patch, then I stay. So there is a couple of assumptions here we deal with. It's an egalitarian group. and they have a cooperative interaction. It's very interesting that whenever we talk about groups, the first thing said usually competition. But I'm gonna treat the cooperative case now, and we have more extensions. But what you do is, okay, you take the same model, but then you add some coupling term to model this coupling, okay? So again, the paper is in the folder. I just want to tell you that we can explicitly then express every type of coupling, but that's what we do with this. Okay, we do the coupling and then compute this probability distributions we have seen and see how do they vary from the non-interacting case. You remember how the probability distribution looked like? It looked like this. The probability P is to leave the patch looked something like this till equilibrium, okay? Till some equilibrium. Is this understood? Okay, so let's then play a game instead of writing down so many equations. Is at every coupling you can tell me what's your prediction. Okay, diffusive coupling. So diffusive coupling, for example, I am with, what's your name? Leo. I'm with Leo, he's reading my mind, I'm reading his mind, what's the group doing? What will happen to the group? So, will the group be more cohesive? Will the group be more, so let's try to look the effect on two things. The accuracy of inferring the rich patch and how cohesive the group is. The cohesion will increase, decrease, increase. Exactly. So what you can, and the accuracy will also increase. So let's go to the, I don't know whether I put the diffusive case first, but let's jump to the diffusive case first. Okay. So the accuracy, okay, it increased, but it's stable over time. But we can see is that if we compute this, the probability, this probability distribution, it basically, it's more spiky compared to, there is more oscillations compared to the non-interacting case. The same with the other distribution. Okay. Let's look at another coupling. The reward coupling is the cohesion decrease, and why? Because you have the single animals. I see you eating. I eat, so there is more probability they stay where they are, so the movement will decrease. So they rapidly, basically converge to the equilibrium. This is the strongest coupling because it's a very direct coupling. Okay, and the accuracy increase. The accuracy here by nature increase because you are assuming cooperative interaction. This is a diffusive. The pulsatile coupling, okay. If the pulsatile coupling, if I am leaving, I leave with you, then I'm more impulsive. So my accuracy will decrease, but the cohesion, of course, will increase. And the reverse for the arrival here, which you can see here, this is the arrival coupling, and then we make the departure coupling 0, the arrival coupling with some values, the cohesion increase and the accuracy increase. And my accuracy decrease with the departure because the animals become more impulsive on average. You can also do this exercise with the counting. Okay, because it's cooperative, I see more animals in the patch, I'm more likely to stay. So the cohesion of the group decrease and the accuracy increase. So you keep playing these games and see, okay, the most important observation we had is this type of oscillatory dynamics. And that by changing the information sharing mechanism, we are able to change the cohesion of the group and the fraction in the best patch. Okay. You can then think, okay, so I'm not gonna, just for the sake of time, and I want to just have some discussion afterwards. We have a paper that just uploaded last week, is then you can relax the egalitarian assumption and begin to have dynamics like, okay, I have a leader and a follower. So how do we model a leader? For example, I can assess the rewards. First of all, you can have misinformation. So you don't share, the leader don't share faithfully the value of the rewards. This is lying or sabotage in terms of anti-diffusion. So means I am accumulating tower threshold, but they try to sabotage, so decrease the, like delay you from accumulating your evidence to reach how accurate this patch is. And yeah, it's cool because you can then take this type of mechanism and see, okay, we found the most important result that I really like is that misinformation specifically will definitely decrease the accuracy of the group, but will increase the cohesion significantly. So that's have a lot of applications in order to mechanistically try to understand things we see on social media and other things. And we have a project with Lisa where she's trying to analyze human data using this framework. Okay, so and why it's also interesting because then the animals have a spectrum of hierarchy. So you rarely find egalitarian animals. You can have a spectrum of hierarchy. I think we talked a lot about marmosets. Marmosets tend towards it's not egalitarian, it's hierarchical, but matriarchal, where female are dominant, but there are very, very hierarchical structures, specifically in Japanese macaque and other types of capuchin monkeys. So the idea behind this is to have a flexible framework where we can try to understand as many animal species as possible. So, okay, extensions in process. So I put this scribble there because that's in 2018 when I began to think about how we extend the model. So one can tell me, okay, so okay, Ahmed, where do you stop? You put more social stuff, and then let's put space in the spatial dynamics between patches or inside the patches. Let's look at continuous environment. Yeah, it's a good question. So we have active projects in terms of extending this to the continuous environment. So a lot of people will tell you, okay, but maybe the animal doesn't really deal with the environment as a continuous way. So let's say, as an example from C. elegans, for example, or larval Drosophila, they, for example, have they do gradient sensing and other things. So can we extend this evidence accumulation model to these cases? Yes, you can by sensing some local gradient, but also at the same time I feel the urge that, okay, where do we stop? And always, like in physics, you are hunted by the question of, okay, you can have this model, they have so many knobs, this detailed mechanistic model, like the ones I'm developing, but then at the same time you have this collective model, some of them are collective motion models, like for people, aficionados, like the Vishek model or the Ian cousin models. So how can we really relate both with each other? From time to time, I think about this. So for example, you can take some of this detailed mechanistic models, try to coarse grain them and see, okay, can we get something similar to this collective models at a larger number of n? Just to tell you what type of problems I'm thinking about. Okay, so what do we do with these models? So Daniel said that, a part of my group is also doing some experiments also along with my collaborators. So that's something that is the most underdeveloped because we took a lot of time to develop these models. But let's say, okay, these are capuchin monkeys in Panama, collected by another group in the Max Planck Institute for Animal Behavior. So what you see here is 10 kilometers by 6.5 kilometers. And these nasty capuchin monkeys. And they are carrying, these are GPS tracking. And the purple here is the resources. And this is the trajectory of a single animal. So I begin to play around a bit with this data and say, okay, what can we learn from, if you have this group of 11 capuchin monkeys from Panama, we can look at the foraging curves, so how how much food they eat over time. We find difference between the dominant individuals, the subordinate one, and then you might, wonder how do we reconstruct the social networks, because we can count the number of either grooming or fights, and we can score the fights and know, the most dominant is the one that is fighting a lot, and the grooming is basically considered an affiliative social contact. But also, like Daniel presented in his talk, to develop a model is something, but to try to fit this model to that is a whole other beast by itself. So we begin a bit of steps toward doing this first by doing some model identifiability, and then looking, how many trials do I need in order to properly infer some of the parameters of the social evidence accumulation models that I presented. And you find, we find 100, but I think it's best if you have like 500 trials. And this is a problem with field recordings, is that somebody goes to the field, they have some data, but you cannot tell them, please get more. So that's still very preliminary work. Another thing is to be able to design experiments inspired by these models. And usually, like I said, you have some controlled lab experiments like I presented in the beginning of my talk. You have the animals either head fixed or in operant chamber, or you can have field experiments that are totally uncontrolled. And in Konstanz, we have this imaging hangar, which is a very large imaging facility where we can try to have large-scale experiments that approximate, you want to have some control, but in the same time, you try to approximate some of the natural features that facing the animals. So one project that my PhD student Maji is doing is using locusts. So what you see here is the locusts are very slow. So these are 155 locusts. They are individually barcoded, and they are supposed to be moving in this foraging environment. And what you can see here, I'm just give you the last clip of the experiment, is this is a high yielding patch and this is a low yielding patch. So basically what we do the design of the models that I presented, and we can have this over time. So you can have them individually tagged, and then you can go on, try to fit this evidence accumulation models and see, are they sharing information or not? Locusts are notoriously solitary. They hate each other, but they team together, for example, in order to exploit resources. There is another project with Daniel, Takahashi, and Paolo, and Ricardo in Natal, where we do the similar also to patch environment with different resources, and we have these two marmosets that are exploiting the resources, so you can, again, do the tracking. There is a lot that goes into this. Like, here, if you look at the locust, we have to individually track them. The same here with the marmosets, and that takes a lot of time. But the nice thing is that we already have some ideas or predictions, intuition from the models that we have developed. We have also a project with humans playing in VR. This, my postdoc Lisa is doing it with a group in Berlin, where humans are playing, are exploiting resources, like collecting fish in this virtual environment, and then you can manipulate the social information by letting them see what the others are doing or not. So we should have a paper out soon. Last but not least is something that I have lately discussed with Daniel is, there is a very smart Israeli student, Tal Nahari. I was giving a lecture in Israel, and she told me, she has saccadic eye movement data where humans will have like familiar, let's say you have a very familiar face here, non-familiar, less familiar, and unknown totally face. And then the saccadic eye tracking is very high resolution that you can basically get the eye movement over time. So what we we have a deadline tomorrow, but to we're able to formulate this problem as a foraging problem. That's a bit of a stretch of mine. So what we are trying to do is to really recast the problem of this eye movement over this. So let's say if this is very informative, so I stay more time here, and if it's less familiar, it's less time here, and the eyes move more than you can imagine. And then what we find is we can basically cast this as a patch foraging problem. But this is also made me think of, okay, did the evolutionary pressure for food resource search optimization shaped a more general purpose cognitive mechanisms that we might be using across tasks? So that's why I believe that we can use foraging as a framework for quantitative evolutionary behavioral and cognitive studies. One have to be careful, like try to, because I was recently in a foraging conference where, it seems to me like, oh, every decision making is foraging. No, it's not like this. I think there is something there deeper in terms of the pressures that have been put on organism for food resource optimization. I have to finish? Okay, food resource optimization. I'm done. Cool. So, at the end of my talk, I want to advertise two things. So there is a Konstanz School of Collective Behavior. Daniel was a lecturer last year. Did you like it?
Yeah, for sure.
Okay, do some advertisement. It's really nice. We provide financial support, specifically if you're coming from Brazil. It's 3 weeks. It's extremely intense, much more intense than here. But you'll get idea about how we can study collective behavior, beginning from cells up to big animals. The other event is in the United States, in Janelia Farm. It's a junior scientist work on theoretical biophysics. Deadline is June 10th. You can see I'm organizing a lot of things, but something I really like to bring people together. This one, the format is, theoretical biophysics, so it's more general. It's really people doing, it's basically student-driven, so it will be a whole week where the student will give both a lecture and a tutorial about a topic, and we basically include topics from people doing molecular things, cells, tissues, animals, full animals, development, but we try to exclude the neuroscientists because just too many neuroscientists around. Okay, so at the end of my talk, I want to acknowledge, the many people I worked with over the years, and funding agencies, and thank you very much. Okay, now
thank you, Ahmed. So, questions?
Any questions?
Well, we had a lot of questions.
You're tired.
Everybody is tired. And,
You said that you were comparing it with experiments by using monkeys. I wanted to learn if you got any results about the capacity of the monkeys to adapt to the environment, if they are as capable as the models, or if they maybe use a different learning strategy.
I have no still insights, I can say. All the fitting and understanding efforts is still at its at its beginning. And then I just put part of the projects we are.
Okay, that's fine. Thank you.
But the advantage of monkeys is that they have clear social structures, and there is a long history of studying them in anthropology or in biology. Yes.
Thank you, Ahmed, for the lecture. I have a simple question, actually. Did you do our simulation in 2D?
Yeah.
Yeah, in the plan? Yeah. And another question. Our stochastic equation you did show.
Yes.
I try to understand what happened there. We haven't when spring-like term to maintain the group together. Another one is a stochastic term, like a Gaussian white noise, something like that. Okay.
Exactly.
And in the middle, you put third one term, which is to correlate the different groups, coupling actually different groups.
Yes.
Okay. Did you have some videos about the simulations? You can show me
I can
after.
ask you for sure. No, I can definitely ask. Yeah, I can definitely get the
Thank you.
simulation for
That's it.
Um, so yeah, I think the model is flexible enough. So we have a collaboration with a group that does slime molds, and you can, amoeba. Yeah, you can still, even on a cellular level, try to, because back to the question I think that was posed about cognition, and I believe that cognition can go down to a cell. A cell is foraging its environment and taking inputs and output, but you can define it as a set of rules that compute something about the environment. I don't think it's a human-only construct.
So actually, I have a question. So do you think that like using your model, you can derive like a generalization of the optimum value theorem, like
Yes.
something that you?
Yes, but I'm very careful with this because I will be attacked. Yes, we are trying, but I don't have anything for now. So a bit of generalization in terms of, okay, if we have, we want to have a generalization that also includes the social aspects of this marginal variation. But I will be very careful because it's just, it's again this picture of the multiple knobs to the small number of knobs. I don't want to fall in the category where, okay, V-check model is very nice, you have a couple of parameters, but at the end of the day, it's really not realistic. And many systems deviate from it, but in the same time, how much is much when it comes to mechanisms? So.
Okay, let's thank Ahmed again.
Thank you very much.
Okay, everyone, so this was the